What is life? Where did we come from? Water is H2O, 2 atoms of hydrogen and 1 of oxygen, pulled together by covalent bonds. The air we breathe is a mix of countless particles, along with dust and water vapor. Then what is it that makes up life? What are the ingredients of our fundamental essence? And what is it that separated us nearly 4 billion years ago and continues to separate us today from everything else?
Open any major news source, and what do you see? We see Trump, we see Brexit. But 30 years from now, 50 years from now, 100 years from now, people are going to look back at now, and they're not going to be talking about Trump, they're not going to be talking about Brexit. What they are going to be saying is that now was the moment when, after 3.8 billion years of evolving by the set of rules we call Darwinian evolution of random mutation and natural selection, and now at the dawn of the age of human precision gene editing, with tools like CRISPR, we're recognizing that the book of life can be written.
Cells are the smallest living units of an organism. All cells have 3 things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it.
These 2 long chains are linked together by the nitrogenous bases via relatively weak hydrogen bonds, but they can't be just any pair of nitrogenous bases. If one nucleotide has an adenine base, only thymine can be its counterpart. Likewise, guanine can only bond with cytosine. These bonded nitrogenous bases are called base pairs. The GC pairing has 3 hydrogen bonds, making it slightly stronger than the AT base pair, which only has 2. It's the order of these 4 nucleobases, or the base sequence, that allows your DNA to create you.
When a cell is ready to divide, DNA condenses into structures known as chromosomes.
There are 2 ways to make copies of cells and organisms. The first and most controversial, of course, is cloning, and that is also known as somatic cell nuclear transfer. And the concept there is very simple. You start out with basically an empty egg, that's the large circle here you see there, and then you place the cell you want to clone, the smaller somatic cell right next to it, and then you send an electrical charge through the unit, and what that does is it damages the membrane between the 2, and the nucleus of the cell you want to clone dumps into that empty egg. And then what you do is you add some chemicals, you fool that unit into thinking it's fertilized, it starts to divide, and you end up with what's known as a pre-implantation embryo. And then you can do one of 2 things with that. You can place that in a Petri dish where you can turn that into embryonic stem cells, which are the master cells of the body, and they can turn into virtually every cell type. And then the other alternative is that you can place that into a surrogate animal to create an entire organism.
Dolly the sheep was a sensation when she first appeared before the world. A scientific breakthrough, fascinating but also menacing. Dolly was cloned by taking a body cell from sheep A, fusing that cell's DNA with an egg cell with its nucleus removed from sheep B. This fused cell developed into an embryo, which was placed in the uterus of a surrogate mother, sheep C. The resulting lamb, in this case Dolly, is a clone of sheep A. These were Dolly's so-called siblings, identical sisters, all cloned from the same sheep, Professor Kevin Sinclair worked with them and knew Dolly.
It was a step change in our understanding of early development, which ultimately led several years later, in 2006, to the discovery that you can actually induce somatic cells. These are sort of cells that belong to the body to become embryonic-like cells that has the potential to divide into other cell types.
Cloning isn't about fusing eggs and sperm in a petri dish, but rather it's about removing the DNA from the egg and replacing it with your own. This would allow you to create an exact biological copy of yourself, or potentially even thousands of copies. However, we must recognize that this has ethical implications as well. For example, if we discover DNA evidence of a clone of you committing a crime, then which copy do we arrest? And how would we know who's who? Some very psychologically disturbing things can happen. What if one of your clones kills you, goes home to your family, and takes your place? Furthermore, if human cloning becomes commercially available, would most people stop having children altogether and just make copies of themselves? Would we ever bother adopting orphan children ever again? Would we bother even getting a date to begin with? We've already frozen the DNA from many people who have died in the past few decades, and we could potentially bring them back. But if cloning does eventually allow the dead to rise from the grave, then could we also create copies of famous politicians, musicians, and brilliant scientists? If thousands of copies of Elvis Presley, John F. Kennedy, and Stephen Hawking are created to serve us, then will there be any room for young people, aspiring musicians, and new inventors to advance? Cloning the past over and over again could potentially bring an end to genetic variation, and thus the end of evolution.